So, hello, greetings, everyone. I hope you are enjoying good health. So, Miss Ilnara, you can start now, please.
Her mic is muted. Miss Nara, Miss Nara, your mic is
Sorry,
muted
sorry,
because
sorry.
you are trying from previous several minutes, but you were trying to say something, but your mic was muted. It was mute.
Are you hearing me now?
No, yeah, we can hear you. It's fine.
Okay, I'm started.
Yeah, go ahead,
Good
please.
evening, everyone, and welcome back to the second day of our international training workshop. I hope you are all enjoyed it yesterday's session and found it informative and useful. Yesterday, we had the opportunity to learn from Dr. Majid Riaz, who introduced us to review articles and shared valuable insights about the review writing process. Today, we are continuing our workshop with Mr.
Sir Ghulam Mustafa?
Yeah, I can hear. I think she is facing some
Internet
network
issue.
problem. Yeah, so she is joining back.
Oh, okay.
We will continue exploring the process of preparing a strong review article with a focus on scientific analysis, organizing information from literature, and developing a clear and effective scientific writing approach. I believe today's session will provide us valuable perspectives and practical knowledge that we can use in our academic and research work. So, without taking any more time, let's welcome Mr. Majid Shahrazad Zafar and leave the session to him.
Thank you so much for giving me this floor. First, let me introduce myself. My name is Mohammed Shahzad Zafar. Currently, I am pursuing a PhD degree in biomedical engineering, Shenzhen University, China. Uh, Today, I'm going to present uh, writing effective results section and scientific graphics in review article. Uh, Good evening, everyone. Today, I will discuss two important aspects of writing a good review article: how we present results or synthesize. Good review analyzes, organizes, compares, and integrates published evidence so that readers can understand the bigger picture. The main message is a good review article should synthesize information, not simply summarize papers one by one. In this slide, here the learning objectives. By the end of today's session, I hope you will understand what we mean by a results section in a review article and how it differs from the results of an experimental paper. We will also see when information should be presented as text, when it is better presented as a table or figure, and how modern tools including AI can help us create clearer scientific graphics. In simple, I want to give an example. The example is that imagine reviewing 50 papers. If you are going to review 50 papers, if we describe all 50 studies in paragraphs, the reader may become confused. But if we organize the major findings into one table or diagram, the pattern becomes much easier to understand. Uh, in If I move forward, research and review article. What is the difference between research versus review article? The figure shows one of the most important differences. In an original research article, researchers conduct an experiment, collect their own data, analyze those data, and then interpret the results. But in a review article, we normally do not generate the same type of new experimental data. Instead, we collect, compile findings from existing studies, analyze them, compare them, and create a meaningful synthesis. For example, if I test a drug on cancer cells in my laboratory, that is original research. But if I collect 100 published studies about that drug and compare their mechanism, effectiveness, limitations, and research gaps, that becomes a review. Uh, Here, I want to give an important message that research articles is what did I find, but review article, what does the existing literature collectively tell us. Next, results in original versus review articles. Here, reports our own experimental findings. And for example, we might report statistical significance or experimental graphs. This belongs to our research article. But in a review article, our results are mainly the organized and synthesized findings from the literature. Original experimental findings, a summary of published finding, like in a review, instead of reporting what happened in our experiment, we explain what multiple previous studies have reported. Statistical analysis, summary tables, and conceptual models. We organize studies according to authors, methods, population, mechanism, outcomes, or other relevant categories. And in experimental graphs, graphical synthesis, we may convert findings from many studies into one integrated mechanism or conceptual figure. And new discoveries, new evidence integration. The value of the review comes from identifying patterns, agreements, disagreements, gaps, and possible explanation across the literature. The goal is not to tell the readers what the individual paper says, but the goal is to tell the readers what the literature as a whole means. If we move forward, then the types of results presentation in review articles. In how to present the result in the review articles, that the some number one, if we go summary tables, useful when we want readers to compare many studies quickly. For example, author, year, methodology, sample size, and major findings. And mechanism diagrams, useful for explaining how a biological or scientific process occurs. And workflow charts, these shows a sequence, for example, literature search, screening, data extraction, analysis, and review preparation. And conceptual models, it belongs, these connect several concepts into one overall interpretation. And research trends, it these show how publication or research interests have changed over time. And the bibliometric maps, these can show relationship between keywords, authors, institutions, research themes, or citation networks. And infographics, these simply complex information into an easily understandable visual summary. Why scientific graphics matter? Scientific graphics are not just decoration, they are form of scientific communication. When readers see several pages of text explaining a complicated mechanism, it may take considerable time to understand it, but a well-designed figure can can sometimes communicate the same relationship within seconds. It improves the reader understanding, increase citation, attract journal editors, and simplify complex mechanisms, improve retention, enhance visual appeal. A strong figure may also attract the attention of editors and readers because they can understand the central message more quickly. If a reader looks at your figure for 20 to 30 seconds, they should understand its main message. Characteristics of a good scientific figure. This checklist gives us 7 characteristics of a strong scientific figure. Number 1, simple. Do not try to show the entire review article in one figure. A figure should normally communicate one central idea. Number 2, accurate. Every arrow, symbol, pathway, label, number, and relationship must be scientifically correct. Number 3, self-explanatory. A reader should be able to understand the basic message of the figure without continuously going back to the main text. Number 4, consistent colors. Use colors systematically. For example, if blue represents one cell type, do not suddenly use blue for another concept elsewhere in the same figure. Number 5, proper labels. Axes, abbreviations, pathways, groups, and symbols should be clearly labeled. And the number 6, high resolution. Images must remain clear after publication, zooming, or printing. The resolution should be according to the journal's specified resolution characteristics according to that format. And number 7, journal compliant. Before submission, always check the target journal's requirements regarding figure, dimensions, file format, resolution, fonts, and other specifications. If the background of your review article doesn't match with the journal, then it should not be submitted. A good scientific figure should be simply accurate, readable, consistent, and understandable by itself. Common types of scientific graphics. Here, the scientific graphics vary depending on research field and types of information being communicated. For example, biological pathways shows biological process from one state to another. Gene regulation shows how genes are activated, inhibited, or controlled. Molecular interactions shows how proteins, genes, metabolites, receptors, or other molecules interact. And experimental workflow shows the major stages of an experimental or analytical procedure. Signaling pathways shows how a signal moves through receptor and downstream molecules to produce a cellular response. And systematic review flowchart shows how studies were identified, screened, excluded, and finally included. Heat map useful for showing patterns in light datasets using variations in intensity. Network diagrams useful for showing relationship among multiple interconnected factors. First, decide what relationship you want to communicate and then select the graphical format. AI tools for scientific graphics. Uh, there are several tools. We have mentioned some prominent tools that are being used in this era. Uh, Today, today researchers do not necessarily have to create they do not have to create every visual element manually. A number of digital and AI assisted tools can help. BioRender are very useful for biological and biomedical illustrations. Napkin AI useful for converting ideas or text into visual concepts and infographics as well. Canva good for simple infographics and general visual design. Mindigraph useful particularly for scientific and medical illustrations. Adobe Express can help with the posters and visual communication. Microsoft Designer useful for creating quick graphics and design concepts. ChatGPT is very famous, can help us plan a figure, organize information, develop figure concepts, improve label and drafts caption. Uh, but AI should assist scientific communication. However, the researcher remains responsible for scientific accuracy. Every generated figure should be compulsory and adhere to the scientific accuracy. How AI helps researchers? AI can contribute at several stages for figure development. Suppose I have written 3 pages explaining a complex signaling pathways. I can ask AI to suggest how this information could be organized visually. For example, if I say that I have designed a pathway, if I will ask the AI what to do, the AI will suggest me the stimulus about the stimulus receptors, signaling molecules, cellular response, and clinical outcomes. And this gives me a starting structure. AI can generate figure concepts, help organize complicated information, assist with graphical abstract, improve caption and labels, and significantly reduce the time required for initial design. So these are the mentioned list how AI help researchers. Never assume an AI generated pathway, molecular interaction, citation, or label is scientifically correct without checking the literature. Never trust the generated pathway as correct. Tips for better scientific graphics. Uh, these are some very simple rules, but following them can dramatically improve a figure. Use simple colors, use a limited and meaningful color palettes. Avoid overcrowding, uh, remove anything that does not help communicate the main message. Main consistency, keep the same fonts, arrows, symbols, line, widths, and visual language. Label every component. Readers should not have to guess what a symbol means. High resolution. Uh, Always export the final figure in sufficient quality according to journal's requirement. Common mistakes. Sometimes a figure contains scientifically correct information but is still difficult to understand because of poor presentation. Too much text. The causes are one of them is too much text. Keep text concise and readable. A scientific figure should not become another paragraph. Poor image quality. Use high resolution images. Pixelated images immediately reduce professionalism. Inconsistent font. Use consistent fonts throughout. Different font styles and size make a figure look unorganized. Too many colors. Limit colors to improve clarity. Colors should communicate information rather than create distraction. Missing legends. The reader must know what colors, symbols, line, and abbreviations represent. The legend should be clear and complete. Unclear labels. If the readers have to guess what an arrow or object means, the figure needs improvement. In short, complex science does not require a complicated figure. The best scientific graphics often make complicated information look simple. Thank you. And any question? But to conclude, I would like you to remember three main ideas from today's presentation. First, results in a review article are mainly about synthesizing and integrating existing evidence. Second, scientific graphics should transform complicated information into a clear and understandable visual message. Third, AI and digital tools can make figure preparation easier, but scientific accuracy and final responsibility remain with with the researcher. Thank you very much for your attention. I will be happy to answer any question. In the end, I want to again repeat an example. The example is, suppose I review 50 studies showing how oxidative stress affects cancer progression. I should not write 50 separate paragraphs saying study A found this, study B found this, study C found this, and that study found that, study C found this. Like, instead, I should identify the common pattern, conflicting findings, important mechanism, and research gaps. Then I can summarize the studies in a table and create one mechanism diagram showing like oxidative stress, ROS, molecular changes, signaling pathways, cancer progression. That is the difference between simply collecting literature and actually writing a good review. Thanks so much for your attention, time, and consideration. I will be happy to answer all of your queries. Thank you so much.
Present question and answer session. If you have any question for our speaker, please feel free and to ask them in live chat. I will read your questions and our speaker will answer them.
Yeah, sure, sure. I'm available.
Please write your questions for today's session. Okay. First question is, can we use BioRender for image generation or any other AI tool?
Uh, in BioRender, you can easily find the biological organs in biorender. The multiple kinds of biological organs are available there. You can generate a figure through AI or some other tools. You need to give the command, the prompt to the AI. It will help you to create a figure, but you must need to draw that figure by yourself by using the biorender or Canva or some other tools that have those biological organs that are easily accessible. One of them, the biorender, and the other one is Canva. Canva is also very, very, very good. I hope I answered the question.
Okay. Can we use images created by AI in review article and how?
And how? Yes, you asked the question that can we use images created by AI in review articles and how? But this depends upon your review paper, how you are going to write, and the pathway, and the story you created, and the literature you read, and you want to create in an integrated figure. Then you have to understand that mechanism, and you give a prompt, a complete mechanism to AI, and it will help you to create the picture. You need to assume that whole mechanism, the whole story. Then you can draw the figure. Suppose if I give you another example, if I want to create a mechanism on cancer, like for example, colon cancer to liver metastasis, how a cancer spread from colon and it metastasizes the liver. It can only be possible when I know the pathway and the background of the cancer. and how it spreads from colon to the liver. I should know the mechanisms, the story, the biological pathway, the interconnected pathway, the cellular pathway. Whole I should know the whole biological route, and then I can easily draw it on AI. When AI suggests a figure, it will suggest a multiple of figures, and I need to collect one, and according to that easily reflects my idea. Then I will select those figure, and then I will draw the figure on biorender or Canva or some other tools.
Third question is, which tool is more efficient to create wide range of grapes?
The graphs you want to multiple, but because my background is biological, biology. That's why I prefer to use biorender and Canva. And after defining the figure, when then I extract it with high resolution. And the tools, there are several tools, but I would recommend, according to my experience, biorender and Canva. The two are more important. And one is perplexity is also good, but perplexity, you can generate the figure but cannot draw. For drawing, uh, Canva and biorender, they both are important.
Here is a question. Sorry to interrupt. Can I
Yeah,
speak? Can I speak?
yeah, sure, go ahead, please.
Oh, here is a question.
Fourth
I
is, sir, I have
yeah.
created a graphical abstract using an AI tool, but after submission, my review paper got rejected because
Yeah,
because of that.
yeah, yeah. I can see her question.
Can I
She is saying
ask?
that her paper is rejected because she created a graphical abstract. First, there are two things I want to highlight. One is that the graphical abstract should not reflect your whole review paper. It should show the central theme. Maybe your graphical abstract is too wide and it is explaining everything of your review article. Maybe this could be a problem here. And the other thing I want to tell you, that if you have created the graphical abstract by using AI, yes, but you need to draw it by using biorender or Canva or some other tools. You cannot submit the same figure that you have created by using ChatGPT or other AI tool. uh, you cannot submit that figure that you have generated by AI, but you have to create to draw it. Sorry, you have to draw it by yourself. Either you draw it by using PPT, or by using by render, or by using Canva, or by using some other tool. You have to draw that picture by yourself. It reduces the AI detection, and when the reader will check the figure, it will make him understand that this is not directly AI generated figure. He draw this figure, he or she draw the figure by himself or him or herself. Yes, this could be a problem. The two, maybe your graphic abstract is too wide, and it is explaining the whole review paper, not just the central idea or central theme.
Yeah. Thank you so much, everyone. I just can I little bit uh, explain the question. Like for example, you are using the Chat GPT, so you can just use the chat GPT just to take the idea, like how we can generate the picture or the graphical abstract. uh, like, you, for example, first you give the prompt to AI, like, I want to generate that type of graphical abstract. So it will give you the idea, like, you can generate the graphical abstract just like this. So it will be easier to generate a new graphical abstract from biorender or Canva, but you cannot use the same exact uh,
Figure.
graphical abstract, yeah, take from chat gpt. So chat gpt figure is not acceptable at all, but you can get the idea from that one graphical abstract. That is the
and
point.
and you need to draw it by yourself.
Yeah, you have to draw it by yourself. I think there is no any question.
Yeah,
Here is
there's
a
one
question.
assuming 15 different studies. I don't think so that 15 different studies have the similar question, but you need to find a research gap, a gap, methodological gap, and yes, you can use AI to take. You can use AI to get a concept that what could be a research gap or the gap why these studies are highlighting a specific issue, and the other possible, what are the other possible questions and suggestions you can ask AI, and you can create, but you need to then you need to start it from the very basic, the background, and then the gap. And yes, you can generate a figure by giving it a prompt. Sometimes AI also suggests the possible pathways, the possible ways, the possible solutions as well. You can give that prompt to AI, and but you have to verify through the literatures that is that really that gap exists, or it forged. You can do it, but first you need to study those literatures and the possible solutions, the gap, and you can predict a figure, the possible figure. It is possible, you can. Yes, if you want to start a review paper, you are a beginner, and first you need to define what kind of a review paper you are interested, and you should uh, design a title. You should finalize. You should final a title, and what you want to write, and then you have to study multiple of literatures, minimum 15 to 20 minimum. That will give you an idea, and you can integrate those idea in one single main theme, and you can highlight the issues, the possible future solutions, and limitations, and in the end you can give a conclusion. But if in the body paragraph, you will write about the everything of the thoughts of that topic, but not in very comprehensive form, because you should also remember that in every journal they specify the word limit, and sometimes some journals also say there is some reference limit. Suppose some literature says 80 references limit and 100 limit. 100 to 80 to 100 are acceptable. Sometimes some journals like cancer letters, they have a limitation 4,000 of word count. Then if you study multiple of the review paper, you have to write a very very concise review paper. And some journals they have 10,000 word limit, and according to that you have to write your review paper. And first you will give the introduction of the topic, then you will start writing your body paragraph, the methodology, and you will explain the further things. And in the end you will give the also you have to give because the editors like you have to give highlight the limitations of that study and the future perspective. And in the end you need to conclude the whole your review paper. And in the introduction, in 3 to 4 lines or sometimes 5 to 8 lines, you have to write that what is the purpose of your paper, this review paper, what is your review paper also going to discuss, because the reader can understand. Yeah. Any more question? Is it ethical to download a sample? I think if you download the same paper from the other site that is maybe already published, but this will be forged and it will be detected, it will have the similarity index. But you can write the same, if you want to write the same paper, you have to give a research gap, a reason why you want to write, what is the novel, what is the novelty in your review paper. That's if you have that kind of novelty, the idea, the different idea that has never been discussed in ever in any of the literature, yes you can write, but you have to give a novel thing so that the editor, the journal can easily accept, and the editor will think that this paper will increase the readership. It is mandatory to explain the figure in theory in an article. It is mandatory to explain the figure in theory in an article. Uh, yes, it is mandatory. You have to explain the figure as figure legend. If you read any review article or any or even any research paper, they explain the mechanism. If you find any review article, in every figure they give the figure legend. In the legend they explain everything, every single step, but concisely. It should be concise. Yes, it is mandatory because when any if I am a reader, I want to study a review article. If I find some figure, interesting figure in it, and yes, they should be mentioned in theory like a figure legend, so that I can understand that figure. Otherwise, how can I understand what is your literature is going to describe? Optimum resolution for the figure representation. Sometimes the journal asks 300 dpi, sometimes more resolution. If you create in very high resolution, it is also good for you, but the minimum requirement is 300 dpi resolution. Mostly the journals ask the figure should be 300, minimum 300 dpi. Yeah, next question. When studying literature for review, do we need to actually read all the? It is not compulsory you have to read whole 20 or 15 papers. You have to get the idea of the related to your topic. Yes, related to your topic, you will collect, you will get that information, and you will work on your specific direction. There's no need you have to read whole 15 to 20 papers in very deeply. No, you have to just take the relevant part. And the several review papers, they discuss several things, but they are some, every review papers have different directions. Maybe some directions are common in some review papers. Yes, you have to take that common information, not the whole, because the whole information will it will mislead you, and it will take you to other direction. It will detract you.
Thank you all so much for joining us today and for being part of this session. I hope you enjoyed it and it useful and informative. That's all of today's session. We will back tomorrow for the third day's hour training, and we had be very happy to see all again. Have a great day and
Thank
see
you so
you
much.
tomorrow. Thank you so much, everyone.
Thank you so much. Have a good day.
Yeah, thank you so much, Dr. Muhammad Shehzad Zafar, for the insightful presentation and discussion. Further, if anyone have any question, you can ask on email. We will share the email in group, and wish you good luck. Be consistent, and the basic purpose of these trainings are at least you have to get idea, like presentation doesn't mean like A to Z each explanation, but you have to get ideas, collect ideas, and further you have to proceed by yourself as well as a self learning. uh, so where you find any difficulty or query, or speaker or presenter will help out you as well by email if you have any questions still. So wish you good luck and stay consistent. Focus on your learning journey rather than certifications, because uh, all you already have certification in the form of your degree. So if you will not focus on your learning, the otherwise the certificate is just piece of paper. So wish you good luck and have a wonderful journey in future as well. Bye bye, take care. So Enara, please, have you any remarks from your side personally? You can mention and conclude it. Thank you. Thank you again, everyone. We have to conclude it.